The overall goal of this procedure is to observe and quantitate the dynamics of signaling complex formation throughout T-cell activation. First, transfect T-cells with plasmids coding for the fluorescently tagged proteins of interest. Seed the cells over a stimulatory cover slip, and visually track the cells as they descend towards the stimulatory plane. Record the cells throughout the spreading process and signaling complex formation. Then, perform Pearson's colocalization coefficient analysis to show whether a complex, like NCK and SLP76, is stable throughout the activation process.
The main advantage of this technique over existing technologies like immunoprecipitation is that this technique enables the tracking of signaling complex formation in time and space in living cells.
Though this method can provide insights into the dynamics of signaling clusters, it can also be applied to other systems, such as immunofluorescence staining, cell shape index analysis, and live threat imaging.
Demonstrating this technology will be Maor Pauker, a PhD student from my laboratory.
Begin with preparation of the 4-chamber slides.
Make sure not to accidentally scratch the inner surface of the chambers during preparation or use.
Fill each chamber of the slide with 500 microliters of slide preparation solution and incubate at room temperature for 10 minutes. Aspirate off all the liquid. Then, incubate the uncovered slides for 1 hour at 45 degrees Celsius to dry.